Hi, everyone. Welcome to our Monday seminar series. And today we have Camille Testard. She just flew in from Boston last night. And yeah, she comes from Harvard. She did her undergrad in McGill in economics. And then she did amazing work with Michael Platt and Laurent Brent at UPenn. And then she recently joined the lab of Catherine Dulac in Harvard University, where she works on... she recently joined the Harvard Society of Fellows. And yeah, she's here for the whole week. So if you like her talk and you'd like to talk to her, just email her and she'll be around till... when are you leaving?
Saturday.
Friday.
Yeah.
Cool. Thank you, Camille, and looking forward for your talk.
Yeah, thanks, Ahmed. Thanks for inviting me. It's really a pleasure to be here at the Max Planck for Animal Behavior. It's been almost a little bit of a dream for me to come here since I was an undergrad. So I'm very happy to be here. And I'm going to talk to you about my thesis work, which focused on the neuroethology of primate social dynamics. But first, can everybody hear me okay? Yeah? Okay, awesome. Okay, so jumping into it. So last year in 2023, the US Surgeon General, which is a big public health figure in America, announced that the country was suffering from an epidemic of loneliness and isolation. And this really brought forward social relationships and social support to a public health issue. And this has come from a line of investigation which started maybe a couple decades ago now, looking at how much social relationships predict survival in humans. And that in this study from Holt-Lunstein in 2010, where they did a big meta-analysis of epidemiological studies in humans and showed that the effect of social support on survival actually, so the relationship is stronger than other well-known health-promoting factors like not excessive drinking or physical activity. And so this sort of, I think, brings very clearly the importance of relationships for humans. But of course, this importance of relationships for survival is not just true in humans, it's also the case in many other animals, which you all know here very well. And this brought me to this, very broad question, which I think concerns a lot of you also, which is why and how are animals social? And in my research, I tried to tackle this from multiple functional levels, from ecological drivers to neural circuits and computation, all the way down to molecular mechanisms. And in my PhD work, which is mostly what I'm going to talk about, I looked at sort of levels 1 and 2. And then in my current work, I am just starting to look at the molecular mechanisms, which I will very briefly touch on at the end. And the animal system that I worked on are rhesus macaques, which are here, look absolutely lovely, group-living primates that form long-term relationships that are really important for their survival, but they can also be extremely aggressive. Well, it's kind of pixelized. And they have these very steep hierarchies, so everybody in a rhesus macaque society knows where its place is in the hierarchy. And so the agenda today, it's going to be sort of a two-part talk, 2.5. The first part is going to talk about the function of relationships, and especially in the context of extreme environmental challenges. And the second part looking at the neural mechanisms of species-typical natural social interactions in primates. Okay. So a tremendous source of instability for humans and all other animals are natural disasters like wildfires, hurricanes, earthquakes. And what they all have in common, these events, is that they're unpredictable and they lead to sustained and really extreme destruction of the landscape and the resources upon which animals depend. And these events have been increasing in frequency over the last 100 years. This is just a plot from our own data showing the number of natural disasters on the y-axis over time. And this this is predicted to continue to increase in frequency and in force. And so this leads to, I think, this question, which is quite important, which might be, what is the role of social relationships in promoting resilience to these catastrophic events? And to begin answering this question, which is really hard to address because these events are so unpredictable, it's hard to have just the data to look at this. I'm going to take you to this small island off the coast of Puerto Rico, that's called Cayo Santiago. So Cayo Santiago is a primatology field site that was started in the 30s by Clarice Carpenter, which brought 400 macaques by boat from India and then released them on this island. And now there's about 1,500 of them. Each individual is tattooed and has ear notches so that they're much better, so that they're individually recognizable, and they're free-ranging. So they're really free to form groups as they please, to associate, to mate. They really kind of choose how they interact with one another. The human intervention comes from provisioning. So they are provisioned, but other than that, there's no human intervention. And I should say, well, I was just going to say that they are for a certain group of them, we follow them on the ground. Humans, research assistants on the ground, follow them daily and record their interactions, what they do on a daily basis. So in 2017, Hurricane Maria hit Puerto Rico, and this was the worst natural disaster in Puerto Rico's history. It led to 3,000 human deaths. And the eye of the hurricane actually went over the island, Cayo Santiago, where these monkeys live. So here are two videos, one from before the hurricane and one from a few days after, 2-3 days after. And as you can see, the island is really destroyed. The vegetation on the island was destroyed. The infrastructure that was there was part of it was blown away. And so we're seeing a complete destruction of the ecosystem in which these animals live. And at first, before this was shot, the researchers thought that probably most of the monkeys died. But actually, that's not what happened. So here you're seeing the death per 100 adults on the y-axis over time. And I compared death rates after the hurricane to a decade, actually 2 decades before the hurricane. And the Shigella outbreak here was in 2010 and was much worse for the population than the hurricane, which led to this small peak here in deaths right after this red line, which is the hurricane. But then in the following year, the death rate is sort of what you might expect based on previous years. So there's an absence of mass mortality. Which is very rare, of course, in animal populations. Usually when there's a disaster of this amplitude, most of the individuals die. And so to understand how the social dynamics change can be quite complicated. So we have this kind of unique, you could call it, setup where we have this free-ranging animal population. Most individuals survived, but now their landscape is completely changed. So here is a plot showing greenery index, which is from satellite imagery. It's a way to measure vegetation over time. And as you can see from the hurricane, there's about a 70% drop in vegetation, which is sustained. And not only is it sustained until 2020, it is sustained until now. And it is really not getting better. Here are videos from images from Google Earth, where you can see 7 months before Maria, there's still trees around. 2 weeks after, the trees are still there, but most of them are dead. And then 4.5 years later, and I would say until today, actually, there's a desertification of the island. So most of the trees are actually completely gone. And it's really a struggle from the field sites to try to rebuild some of that vegetation. And then they're just they're not succeeding at doing that currently. And because this is the Caribbean, temperatures can get really high. So we have temperatures loggers across the island in exposed areas and shaded areas. And this is showing just the average temperature over an entire year, I think it's from 2018 to 2022. And on average, you can have daily temperatures up above 40 degrees Celsius. And the only way, or the main way, that these monkeys thermoregulate, that they lower their body temperature, is by accessing shade. So as you can see, in the afternoon, there's about on average, an 8-degree difference in temperature. But as I showed you before, the vegetation is really destroyed. So shade is now this really scarce resource that these monkeys are struggling to get access to. So here you have, for example, an image of an old low-ranking male that was following around this research assistant to get a little bit of shade. And here, these monkeys lining up, I think this was around 9 a.m., behind this dead tree to get a bit of shade. And so together, this leads us to ask the question of how did the rhesus macaques adjust socially to this drastic and sustained change in their environment and to this sustained exacerbated heat stress due to stronger exposure to heat and lower shade resource that they use. So this is a longitudinal data set. I've used data from 10 years on 7 social groups, 790 unique individuals, and about 250 of them that we have data really pre and post hurricane. And we've collected data using a combination of focal follows, which is when you follow an individual for a set number of time and record everything that the individual is doing, or group scans. And we've collected data on 3 different interaction types. Grooming, which is an active form of social investment. Proximity, so whether these monkeys are sitting next to each other within 2 meters of each other. And finally, aggression. So all of these 3 behaviors, if you will, tell us something about the social structure of these animals. And this was not collected with drones or with GPS tags or any of that. It is old school humans that have collected, mostly Josue and Danny, that have collected this longitudinal data, as well as other people on the ground. Okay, so the first set of results, which maybe you might expect, is that there is a huge change in the proximity network. So animals are spending way, way, way more time in proximity to one another than they used to. And I should really insist on the fact that these are rhesus macaques who are extremely aggressive, and they really choose who they sit next to and who they allow next to them. And as you can see those from the social networks, each node represents an individual, each line is a proximity interaction. There are way more proximity interactions. So individuals spend time next to way more partners than they used to. And then here you're seeing the probability of proximity, and I'm plotting all of the individuals we have data for, and there's a sustained increase in the proximity. We did not see a sustained increase in grooming interaction, however. And this sort of leads us to ask, are these proximity changes indicative of just indicative of a spatial preference for shade, or do they really reflect a social change, a change in the patterns of interaction and how they consider other macaques on the island? And the first piece of evidence that I think it is the latter is that there's a sustained decrease in aggression. So even if we're talking about a very aggressive species that can violently contest resources, and that there are less resources, that the shade is scarcer, we have a sustained decrease in aggression. Not only that, but if we remember here that in the morning, there's not a very big difference between being in the shade and being exposed. So in the morning, in a sense, monkeys can sit wherever and it won't make a big difference. Yet, if we look before and after the hurricane, and we compare in the morning, this is just the average probability of any monkey being next to any other monkey. There's about a doubling pre-post hurricane in the morning and a tripling in the afternoon. So even in the morning, where sitting in the shade is not necessarily useful or necessary, there is a much stronger or higher probability of being next to other monkeys. Finally, not exactly finally, but so monkeys, if we look at proximity events, about 40% of them do happen in exposed areas. Those happen in the morning. And in the afternoon, they happen in the shady areas. And the two are highly correlated. So whether I sit a lot next to other monkeys in the morning predicts whether I sit a lot next to monkeys in the afternoon, and vice versa. So proximity between monkeys, I think, really reflects a social change in social decisions, not just a preference for shade. The next question, now that we have more years after the hurricane, we can start asking questions about the fitness consequences. And so we asked whether this social flexibility have consequences for individual survival. And we ran survival models controlling for age, sex, and social status. And here I'm going to show estimates. Estimates that are less than 0 means a decreased risk of mortality, and above 0 is an increased risk of mortality. And I split the data into pre and post-hurricane. So pre-hurricane, whether you spend some time next to other monkeys or the number of monkeys that you sit next to doesn't predict your survival. Doesn't really matter for survival. After the hurricane, however, it does quite strongly. There is, for a 1 standard deviation increase in or higher probability to sit next to other monkeys, there's a 42% decrease in mortality. So it really is quite critical for these animals. And when in the afternoon proximity, so whether I spend a lot of time next to monkeys in the afternoon, where that happens in the shade, that more strongly predicts your survival. So what we think is happening is that the hurricane exacerbated heat stress due to the destruction of the vegetation and the high temperatures. Social tolerance, being next to other monkeys, being tolerated by other monkeys, facilitates access to the remaining shade that's available. And this reduces exposure to intense heat and leads to better chances of survival. And so the big take home here is that by abruptly causing long-lasting changes in the ecosystem, natural disaster can alter the adaptive benefits of sociality, which can trigger persistent changes in the social structure of group living primates. So this is the first part, and now I'm going to switch to the second part, which here happens not in the wild, but in the laboratory, where I looked or tried to ask what neural mechanisms underpin macaques' ability to connect and sort of flexibly adjust their relationships in times of need. And here we used electrophysiology, which is a method where we do intracranial recordings, which require surgery. And we can implant chips that have a certain number of electrodes and actually record the electrical activity of neurons and record action potentials to then relate this to the behavior of these animals. And to really explain why this is new, I think requires a little bit of background on how we've been studying primates and the neural mechanisms of social behavior in primates, including humans, actually. So the only methods really that exist and that are used are either fMRI or MRI-based approaches, or electrophysiology intracranial recordings in animals that can't really move, where we have to basically restrict their head movements, restrict their whole body movements, because otherwise, until very recently, that would completely jitter or add noise to the signal. And using these approaches, we've discovered quite a bit of things about how the primate brain processes social information, like facial expressions, vocalizations, also how neurons encode my actions versus the actions of another, or even quite advanced levels of interactions where you might have two monkeys playing a cooperative or a competitive game through a sort of a touchscreen based or sort of indirectly to some extent. Well, directly interacting, but via a game. And it had not been possible until recently to actually study their natural social interactions in freely moving animals. And I think this is quite important because if I take a very quick analogy, let's say we want to understand what a car is, and we put the car on a pedestal and we start poking at it, which is a bit what we're doing with monkey brains. And you hit the radio button, and then the car is going to blast some sort of music. And that might be a source of an indication that actually a car is a sound-producing machine. And you might try to interpret other findings relative to this one initial finding. But it's only if you take the car and you put it on the road, which is kind of what it's been designed and structured to do, that's when you start really understanding what it's for and how the different components come together. And I think this is why we need to study the brains of animals that are really moving and performing natural behaviors. Okay, so here is the technology we've used. It's called a data logger technology. It is about 33 grams. And it's basically a computer that's all within this tiny box and that is connected to a little chip here that you show here. This multi-electrode array that has up to 128 electrodes. And here we are recording from hundreds of neurons while these monkeys are completely free to move and actually interacting naturally. And this is work with Sébastien Tremblay at UPenn. And using this technology, we were able to record neural responses across macaques' natural behavioral repertoire and different social contexts. And I should say, of course, it's not the entire behavioral repertoire. It sort of recapitulates some, I think, quite fundamental aspects of how macaques interact with one another. And the experimental design was in the home enclosure of these animals. We wanted to least disrupt them as possible. And we recorded from 2 males in 2 male-female pairs. So here the subject is represented in green with the little neural logger. And then the male subjects were paired with a female partner. And there was a separator such that we could have the animals paired or separated and the subject be isolated. And then when they were paired together, the subject had visual access and could interact, just not touch, but could interact with a neighbor. And that's the neighbor's cage here, where this is another male-female pair. And we would close the separator here, and we could switch around who is the neighbor, whether it's a male or a female. Okay, so during this, and this was data recorded with Felipe, who's a very talented grad student in the lab. And during these experiments, the monkeys would perform a whole bunch of different behaviors from grooming. There are a lot of reciprocal grooming, foraging, traveling, just moving around the cage, approaching the partner, leaving the partner, aggressive interactions, mostly with either a human experimenter or with neighbors, and yeah, self-grooming. So a lot of different behavioral states, which again had never been explored neuroscientifically until now because it had not been possible. And we recorded in 2 brain areas to just give you a very brief overview of, or like sense of why we chose these 2 areas. Usually the sort of social information processing pathway in the primate brain is, there's sensory input that goes into, for example, B1, which is the primary visual cortex, or A1, which is the primary auditory cortex. And then these sensory cortices, the information first goes there and then gets sort of sequentially processed into more and more complex visual stimuli. And area TEO is sort of a mid-level visual area. So you have, up until now, had only neurons described to respond to certain angles and certain lines of the visual field. And then this visual information and auditory information is going to be sent to the prefrontal cortex, and we recorded in the ventrolateral prefrontal cortex in particular, where this sensory information is combined with other types of signals, internal states, and sort of more decision-making related signals. And so we recorded in these two very distinct brain areas, one that's more on the side of input, visual input, and one that's more on the side of the output and the decision-making, because we wanted to get a sense of where certain types of information might be located. And we had this one prediction that the neural activity would really vary according to the behavioral state and the social context and these more complex variables, behavioral variables in the prefrontal cortex, but much less so in a visual processing area. These are just two 3D reconstructions of the monkey brains of our subjects, and you see where we've implanted, and we recorded on average 300 neurons per session. Okay, so getting into results. Here I'm showing the activity, the firing rate, so the number of action potentials per second of an example neuron over time. And I've signaled where the blocks are, the different social contexts. And now I'm going to overlay what the animal was doing at any given second. And you might notice that whenever the monkey is grooming his partner, there are pretty consistent dips in firing rate. And when the monkey is being aggressive towards his partner, there are these peaks in firing rate. So there's pretty consistent relationship between the activity of the animal, the overall behavioral state, and the neural activity. And this is, notice, a TEO, so visual. TEO is a visual, this visual area, which was already not super expected. And now I'm going to show you sort of an overview of the activity of all the neurons that we recorded from in an example session. So each neuron is going to be a row. And yellow means higher activity relative to the mean, and blue means lower activity relative to the mean, and you have the behavior on top. And I've split neurons by TEO on the top and in the prefrontal cortex on the bottom. And what you might notice, just kind of eyeballing it, is that there's sort of a blocks of activity. So certain population activity that are quite characteristic and differ according to the behavior of the animal. And this is really true across both brain areas. Now, if I represent this activity in a slightly different way, rather than looking at each individual neuron separately, we can look at the combined activity of all the neurons. So if you imagine that your brain has 3 neurons, you can represent the combined firing rate of all of them and your neural population state by a point in that 3-dimensional space. We have a hard time imagining more than 3 dimensions, but a computer can deal with that. And so here I'm going to show plots that are going to show these different neural population states, and dots, points that are close to each other means that the neural activity is more similar, and when they're further apart, that means that the neural activity is more different. Okay, so we used an unsupervised nonlinear dimension reduction method called UMAP, which is quite standard becoming quite standard in neuroscience. And here, each point represents the population state of the neural population state for 1 second. And as you can already see, there's clustering happening. And now I'm going to color code according to what the animal was doing at any given time. And we find very clear behavioral clustering clustering of the neural population activity by the behavior of the animal. And this is true across both brain areas, as you might have expected from the previous plots. But you might also notice that there are 2 groom partner cluster, for example, or 2 getting groomed. And now, instead of color coding by behavior, I can color code by the social context. And then these separate clusters make a lot of sense. So this top one is I groom my partner and my neighbor is a female, versus I groom my partner and my neighbor is a male. So in other words, there's, in both brain areas, very striking and, this is my conclusion, conclusion, very strong modulation of the activity according to the behavioral state of the animal and the social context in which that happens. And now I'd like to zoom in on a particular set of behavior that these monkeys do, which is grooming, and in particular, reciprocal grooming. So this is one way that animals, say of similar rank or siblings, will form and maintain strong social bonds. One will groom each other, kind of the same as in humans, where my friend, if it only goes one way, you know, we're not going to be friends for a very long time. So it has to go both ways, right? And this reciprocity, we see this in the wild, and we also saw this in the laboratory. So both of our male-female pairs were very, very reciprocal in their grooming interactions. So on this left plot, you see number of grooming bouts given in blue, in cyan blue, light blue, and received in dark blue. And so for both pairs, you have very close number of grooming bouts, and also grooming time, the number of seconds. And for this pair, for example, out of almost 20,000 seconds of grooming over a month and a half, at the end, there's about a 50-second difference in how much they groomed each other. So they really reciprocated these grooming interactions. And this reciprocity happened over the course of a session, right? There was some reciprocity within the course of a session. But at the end of a session, you might find that there's a bit of a deviation from equality, from perfect reciprocity. And if you look across days, what we found is that whenever on one session I groomed my partner, or I received more grooming from my partner than I gave, on the other session it was the opposite. And they sort of, over time, it would would equalize. And so with a bunch of different simulations, we're able to show, and we are able to know that the only way that they can obtain this sort of equality is by actually keeping track of it and really wanting to be equal. And so this led us to try to investigate how neurons might be tracking reciprocity. And so here are just two example neurons, again, from both brain areas, where you can see very clear modulation according to whether I'm grooming my partner or I'm getting groomed. And we can use information or we can look into just the neural activity and use different decoding approaches, and I use the ridge regression model to see whether I can predict from the neural activity alone whether the animals, the level of reciprocity during a session. And so here's the model prediction, again, only based on neural activity of the level of reciprocity over time. And here is the actual level of reciprocity. And so essentially across sessions from the neural population activity, we, in both brain areas, so both either or, we can extract much more than chance what is the level of reciprocity. And one other, I think, last interesting observation is that here I'm showing again these neural population states. But now I'm showing just for grooming on the top and rest on the bottom. So rest is just when the monkeys were sort of sitting and looking around and not doing much. And if we look inside of these grooming states, and now I color code according to the bout number, like where are we in terms of how many bouts of grooming have happened, there's a clear structuring of the population activity by time, which is not there, or very less clearly the case in rest. So that means that, and we've also used decoding to show this, that there is a stronger tracking of time during grooming than in other behaviors, which I guess kind of makes sense when you think of, if I'm trying to be reciprocal, I need to keep attention and make sure I keep track of how much I'm giving and receiving, versus when I'm just resting, it doesn't really matter how much time has passed or how many bouts of rest I've done. Okay, so this brings me to the take home number 2, which is that neurons really across the cortex, even in sensory areas, which was unexpected, are strongly modulated by natural behavior and social context, as well as reciprocity of interactions. And we find a distributed in the sense that really across very different cortical areas, neurophysiological record of social dynamics in the macaque. Okay, so if I come back to my original question, why and how are animals social? Today I've talked about how this hurricane altered the adaptive benefits of being social and led to these striking changes in the pattern of social interactions in the Cayo Santiago rhesus macaques. And I think shows that this sort of social flexibility might be a really important asset for these animals to adapt to these kind of abrupt events. And then I've looked into more of the neural circuit and computation of natural social behaviors in the monkey. And I think, again, these are behaviors that had really not been explored before, so there's a ton more that needs to be done to try to figure out really the computation, the neural circuit and the computational basis of these behaviors. But I think this is really the beginning of a new line of investigation and a new era in primate neuroscience. More and more people are wanting to do this sort of work, which is very exciting. And I think this has really big implications, not only scientifically, but also ethically. I think that the lives of these subject monkeys are way improved with this sort of methodology than what has ever been used before. Okay, so just coming back to my little diagram, I'm just going to briefly touch on my current research, and then that'll be a wrap. So in my current research, I kind of pivoted quite a bit to more rodent models because I wanted to get at molecular mechanisms, and you can very hardly do that in monkeys currently. And here I focused on, okay, so the hypothalamic nuclei. So the hypothalamus is this part of the brain where different circuits have been really identified as controlling innate social behaviors. And people like Catherine Dulac, who's my supervisor, my postdoc advisor, but also other people have shown that specific cells in the hypothalamus and in the medial preoptic area have control in the mouse, mating, parenting behavior, pup-directed aggression, so like infanticide, social drive, so whether an animal wants to be next to another animal or not. And if you sort of take a bird's eye view and you combine all of this, it looks a lot like all these different cells are sort of defining the social system of an animal, which is like, do I want to live in a group or do I live alone? How do I care for my offspring? What is the mating structure? Am I monogamous or polygamous? And there's this hypothesis, I think, that emerges from this, is that in mammals, this medial preoptic area of the hypothalamus might be this sort of physiological knob that controls basic social tendencies, leading to variation in social systems. And all of our knowledge comes from laboratory mice, and we have no idea how these circuits might generalize and might be modulated in other species. And I'm starting with rodent species that are quite different socially. So for example, the African spiny mouse here, which is highly gregarious, they're found in these like large colonies in Southern Africa. Or the African striped mouse, which is socially flexible. So depending on the context, they can be solitary or group living. And what I'm starting to look into is how these circuits, these neural circuits that have been identified and very well described in the laboratory mouse, how are they modulated in these other species? And you might imagine differences in the distribution of different cell populations, differences in the gene expression of these different cells, or in connectivity, or some combination, likely, of all of these things. And so that's my current/next step. And yeah, that wraps it up. So hopefully in the future, with all of these different approaches, we'll be able to connect ecological drivers to neurocircuit and computation and molecular mechanisms to understand why and how animals are social. I'd like to thank the monkeys that have participated and that have, from which all of the data I've talked about today comes from. So the Cayo Santiago macaques, Lovelace, Amos, Sally Ride, and Hook that I've worked with for many months. And then all these human primates that have also helped me along the way, collected the data and supervised me. And and I'm happy to take any questions.
Thank you, Camille. Questions, and I will pause the mic.
Thanks. This is a really nice ride through the various different ways of looking at mammalian sociality. Going way back to your first study, I was wondering, have similar trends been observed in any of the primate species native to the Caribbean?
The primate species native to the Caribbean, that's a good question. I don't know. I don't think that there is any other field site or people that I've studied that have the data really to look at this, or at least I haven't been made aware of this. So I don't know how that's been the case. We do know that there are reports in humans a lot that after disasters, there's increases in social tolerance, in cooperation. So from that perspective, I guess in human primates, yes, but in other non-human primates, I don't know.
Yeah, thanks for the talk. I had a question also about the first study. I was wondering if you looked at differences at the individual level, since you had IDs both before and after the hurricane, but you showed more of an aggregate decrease. And if you looked at that, did you find that more aggressive individuals before were still more aggressive afterwards, or was there
So what I will say is that for proximity, for example, every individual that spent more time in proximity to others. That's really a population-wide effect, but there is inter-individual variability there. And we're only starting to look into this, but, and I don't have the plot here, but what we're seeing right now is a big sex difference. With males actually showing much more change than females, but also males tended to be more peripheral to the network, and those are the ones that seem to be changing the most. Yeah.
And is it just a quick follow-up? Have you looked at dominance hierarchies, trying to quantify them before and after and seeing if there was a reshuffling?
That's a good one. No, I have not. No, we have not. But that's a good question. Yeah.
I have one that sort of follows on from that, but it might be a bit of a big jump, so it could be just open for discussion. But with the tracking of time of how much you give and receive in the grooming, could there be anything about how you use a limited resource that, like the shade, where there could be a tracking of, I've had this much of a resource, you've had this much of a resource, how that could come across in your social activity? I mean it in the way of a dominance hierarchy. Could you be keeping track of other individuals forcing you out of such a resource or allowing you to gain access to a resource?
Yeah, I think that's a very interesting question that I haven't looked into directly in the data. What we have looked into, so I haven't shown this because I didn't really have time, but another big manipulation we did in the laboratory was threat events. And so you'd have a human experimenter, someone external, that did threats to either the subject or his partner. And then we had social support. So we really had a response from the subject when his partner was being threatened. And we had very strong tracking of, very strong responses of the neurons that were very similar, whether the subject was being attacked or its partner. And that's another dimension when we think of like how much I'm giving and receiving. It's not just whether I'm grooming, it's how much I'm also sharing food, also support. And here in this very artificial setting, there's very few contexts. There's very few ways that the animals can reciprocate. And the main way that they can do this here is by grooming. So that might explain why they're so equal. Versus in the wild, support in when there's aggression or sharing of resources, those are other ways that you can reciprocate and that you can actually, feel like or maintain relationship. And so I think potentially, all of these things are being kept track of. And we haven't looked at the combination of these very different ways that animals support each other or might give and receive. But that's really future direction, I think, that would be very interesting. And my prediction would be that, yes, you sort of combine these different contexts, essentially.
Yeah, thanks also for me. Super, super fascinating work. I have a question about the neural side. And I'll try to structure this because there's like many questions layered in one. But so the fact that you saw that the supposedly sensory area as well as the higher order areas both essentially at state-dependent modulation, if that's what it can be interpreted as, with the social behaviors. Very similar to, I guess, how it was discovered a long time ago that you would also have state-dependent modulation with like motor activities, for example. The question is kind of, is that how you think about it? Or do you think about the visual area also having something to do with, like directly to do with the social processing? And then kind of going on from there, how do you figure out the correlations? I guess for the motor actions, you can often have temporal correlations, like is the activity actually preceding or the brain activity preceding the motor activity? Like how do you, if a lot of brain areas are modulated by social cues or social experience, how do you figure out which brain areas are actually like causally responsible for controlling the behavior of the animals?
Great question. I think that you will never figure out which one, what is causally contributing to a particular behavior only using electrophysiology. So this is a big limitation of electrophysiology. It is purely correlational. So you might, like you said, you might look at temporal alignment. That might be a clue, but in my view, that's still not enough to say that that's causally implicated. And I don't think necessarily that TEO is causally TEO, the visual area, is causally responsible for the different behavioral states and social interaction and keeping track of reciprocity and all of that. But I think the fact that there is this information there and that it is represented very strikingly, that tells us more something along the lines of that actually your behavioral state and your social context might influence the way that you perceive the world, right? Because really the activity of the neurons varies so much by the overall behavioral state of the animal that if you have different visual inputs during those different behavioral states, that might be processed or computed very differently. That's still really open questions, though. I think much more studies need to happen to really figure that out. But it tells us that it's much more complicated than just if I stick an electrode and there are responses to, say, visual input, then this is a visual area and it processes like whether this line is this way or that way. I think it's much more dynamic and interrelated with a lot of feedback, right, between the different areas. Does that answer it? Okay.
Can I, just because I have the microphone and I'll be naughty, just a super quick follow-up on what you just said with the bar encoding. Let's say, do you have a feeling for how strongly these modulations are relative to if you would have them groom each other and watch a screen where, for example, you're modulating bars and you're checking the visual area?
See, that great study idea. No, we have no idea. This is the first study ever looking at this. So currently, no, we have no idea. What we do see is, so when you have monkey sitting in a chair doing a visual attention task, for example. So, they're going to be fixating and then there's going to be a cue and you're going to look at responses of neurons to that cue. The neurons fire quite, they respond to this very strongly. And I think to some similar extent to this, but it's such an artificial setting so that what are you really, you're forcing the animal, you're training them for years sometimes to pay attention so that little signal, that 50 milliseconds that you have to see this is whether you're going to get a reward, whether you're going to be able to drink today or not. So obviously the neurons are going to be very, very responsive to this. It's really important. But now in this more natural context, we don't know. And I think that's, would be really interesting to look at.
So Camille will be here till Sunday and
Sunday.
Friday.
Friday,
Sorry,
Friday.
I blacked out. And thank you, Camille, and thank you all for coming.
Yeah, thanks.
Thank you.